Thank you very much indeed, and I apologise for speaking to you in English after what I hope has been a nice, heavy Italian lunch. We've had a wonderful morning, haven't we, with some discussion of the evolutionary concepts and how epigenetics fits in, some philosophy, some of the detailed mechanistic implications, and then the wider applications. And I think it's some of the wider applications that I'll be beginning to discuss with you. First of all, I want to draw your attention to the idea of DOHAD, the developmental origins of health and disease. There's an international society now for the study of these phenomena, so those of you students in the audience, I'd urge you to join. It's very cheap, and you get a journal and information about the activities of the society. What I'm going to do over the next 45 minutes or so is to try to address four things. One is the concept of soft inheritance and how that and our evolutionary past actually echoes into our health today. Then I'm going to say, as of course, because of the meeting, the role of epigenetics, and then end with some of the wider implications. I have to acknowledge two debts of gratitude. One is to a long-term collaborator who works down there in Auckland, New Zealand, on the other side of the world, Professor Peter Gluckman, and he and I, with a colleague from New Zealand, Alan Beadle, produced a book some years ago called The Principles of Evolutionary Medicine. And thanks to the patronage of Gilberto Gobellini from Rome, this book's now been translated into Italian. Well, we heard a little about the modern synthesis earlier on today, and the more one looks back on that and the way that Ernest Mayr and their colleagues synthesized it, the more, to me, growing up as a developmental biologist, I'm astonished at how gene-centric it is. The three tenets, if you like, of Darwinian evolution, that there is variation between individuals, that there's selection of fit individuals, and that those individuals are then able to pass some aspects of that fitness onto the next generation, are all founded in the concept of genes. The variation is purely in terms of fixed genetics. The selection is on the genotype, not on the phenotype. And the inheritance is purely fixed genetics. Absolutely astonishing when you look back at that from the perspective of the early 21st century. Those of you interested in the history would probably do well to read a book by Fox Keller from the States, The Century of the Gene, where she talks about the 20th century and how this genocentric view dominated all our thinking. Although actually, if you go back to the beginning of the century, William Bateson, shown here, who was the person who actually coined the term genetics, he didn't coin the term gene, but he established, if you like, the science of genetics, said, and I quote it here, genetics is the physiology of descent. Well, genetics these days isn't physiology, it's all structural, it's fixed stuff, the genes and mutations in them. It's got nothing to do with all the homeostatic mechanisms that the body employs in physiology. So somewhere we got very sidetracked during that last century. And as always, when you think that there's something puzzling you and you think perhaps you've got a new idea, you go back to Darwin, and sure enough, you find it's there in his writings, even if he didn't necessarily conceptualize it in the way that we do. Look at this quote, which I like very much from The Origin. Let me read it to you. It is well known that animals of the same species have thicker fur and better fur the further north they live. Fair enough. But who can tell how much of this difference may be due to the warmest clad individuals, the ones with the thickest fur, having been favoured and preserved during many generations? So there's no surprise there. That's just natural selection. If you're living in a cold climate and you happen to be born with thicker fur, you're going to be more likely to pass your genes on to the next generation. So that's what we now see as classical Darwinism. But look at the next sentence. And how much to the action of a severe climate? What on earth was he saying? That somehow the environment could alter the way that individuals, develop? This is Lamarckism, isn't it? And of course, many of the books, some of them actually by quite well-known people, sadly, Think of Lamarck and Darwin as being opponents. There are famous cartoons showing them battling it out, each holding monkeys or parrots or whatever you like. In many ways, they didn't battle it out, and Darwin had a lot of respect for Lamarck. He doesn't mention him a great deal in his writings, but he certainly didn't dismiss some of the concepts that Lamarck can advance. And actually, as we heard this morning, the inheritance of acquired characteristics, which we all think of as being Lamarck, actually wasn't really what he said. He's been rather misrepresented. So Darwin was beginning to think in what I'll show to be a heretical way. But it was really, to my mind, the science of embryology in the beginning of the 20th century that started us off on the wrong path. Because embryologists began to think of the genetic program of development, and they wanted to study organisms where the similarities between developmental trajectory were similar. They didn't like all this noise, all the environmental effects that could come in and mess up the story. So they tried to confine themselves to models where development followed a pattern, it followed rules, it followed laws that they'd laid down. And of course, that became enshrined in the genetic program for development and the way that pattern formation and so on occurs. This isn't necessarily helpful in today's world, because it has been known for many, many years that environment is incredibly important in development. This is just one historical example, which refers to a phenomenon called maternal constraint that I want to spend a few minutes on because I want to show you that it's actually incredibly important. This is a study from two biologists, Walton and Hammond, who published it in 1934. And what they did was to mate two different breeds of horse. This is the cart horse, the Shire horse that draws the plough, or did in the 19th and early 20th century on the farm. This is the tiny little Shetland pony that children learn to ride, and it lives on some of the islands to the north of Scotland. So what they did was to mate, in this case, let me get this right. Over here we have the Shire mare, the Shire female, the big female, mated with the tiny little Shetland male. And over here it's the other way round. Here's the Shire male over here, mating with the tiny little Shetland foal. And they wanted to, in case you're wondering, this was the early days of artificial insemination. And the question that they asked was, what would the offspring be like at birth? Would they be, in the Shire Shetland cross, would they be as big as the dad? In the Shetland Shire cross, would they be as small as the dad? What was going on? And of course, as perhaps you can see from the slide, actually the offspring here at birth were much more the size and shape of the offspring of the mother for that breed than they were for the father. And even at 1 month of age, and as they grew up, they stayed in that pattern. So this concept that the mother constrains to a degree the growth of her fetus in order so that it can be appropriate for her reproductive tract is shown very clearly here, and it's been reinforced by many studies in humans. To bring evolution in, let's go back to the future. Let's go back to where we came from. This is the part of the Rift Valley in Ethiopia, where of course 60,000 years or more we walked out of Ethiopia and started to move into Europe and so on. And in this part of the world, even today, you can be a long way from medical care. And so if you look at the evolutionary components that led to our survival, there are at least two that are important. One is, of course, that we began to walk on two legs, bipedalism. That changes the shape, the size and the orientation of the pelvis, so that now, unlike an animal that walks on all fours, it has to support the weight of the guts as we run around the savannah. The second thing is that we involved very large brains, and therefore, of course, very large heads. And if we compare humans at the time of birth with our nearest ancestors, at least in terms of our genomes, the chimpanzee, you can see at birth what a huge challenge we have. Here's the human head passing through, but only just, the pelvis at birth, and it has to rotate to get through. The chimpanzee has absolutely no such problem. So the size and shape of the mother's pelvis has to determine, to a degree, the size and shape of the fetus. And so maternal constraint must be very important in terms of fetal growth. And sure enough, if this process goes wrong, as it does regrettably in a large number of pregnancies, then you can have obstructed labour, you can have rupture, fistulation of the maternal genitourinary tract, and if you're a long way from medical care, as some women in Ethiopia are, you can end up literally having days of obstructed labour, and usually by the end of 3 to 4 days, by the time the woman has walked for a day to find a road, a day on a truck to get to a hospital to find that there's nobody there, no anaesthetist, the fetus is dead and she may well not survive either. It's more complicated than that though, because maternal constraint depends on a whole range of maternal characteristics. It's greater in first pregnancies. You might expect that because the reproductive tract has never been stretched, the blood supply to the uterus has never increased in that particular individual, and so she's going to constrain the growth of their fetus a little bit more. It's greater if the fetus is a male, it's likely to be growing faster. It's greater if the woman is very young, a teenage conception, or if she's an elderly prima gravida, with poor diet and with smoking and so on. And so actually, even the Ethiopian data shows that the days of obstructed labour are even longer and even more risky in a first pregnancy, a prima gravida, than in a subsequent pregnancy in that woman. Well, why am I bothering to tell you all this? It sounds a bit of interesting biology, but surely in a developed world, in 20th century Europe, with proper medical care, these issues aren't important at all, are they? Or are they? So a couple of years ago, we decided with some colleagues in the Netherlands to look to see if we could find any evidence of maternal constraint in a human population. They had access to about 1.2 million births from the Netherlands birth cohort, I think between 1990 and 1998. These were all normal births. We excluded those with congenital abnormalities. And we simply asked the question, what's the best size to be born? In terms of what's good for the mother, in terms of what's good for survival. And is there actually some kind of a conflict there? Because you might argue from this that mothers would want their babies to be a little bit smaller, but babies would want, if you like, to be a little bit bigger. And sure enough, when you look at the Netherlands data, it's absolutely astonishing. Here we have those 1.2 nearly million births. This is just literally birth weight shown as centiles, from 0 here, if you like, down to the biggest babies over here. And this is mortality per 1,000 live births. They're very small babies, they're terribly growth retarded babies. And by the way, these aren't preterm, although the data is the same for gestational age. These ones have a hard time and many of them don't survive. The very, very, very big babies above the 97th centile may well be macrosomic. They may have real problems and their survival is less. So where's the optimal place to survive as a baby? Presumably, with modern medical care, it's smack bang in the middle at the 50th centile. This is the normal human delivery, isn't it? Well, that's here, which might be good for the mum, but actually optimal survival's to the right, with babies bigger than that. And so one way of looking at it is that this echo of evolution, which is playing out in our maternal constraint forces even now, is actually still bringing with it a huge burden of mortality, even in a Western world. So some years ago, Peter Gutman and I came up with the concept of mismatch, trying to put the evolutionary and the developmental components together. And we argued that as a species, we're generalists, we've been able to adapt to a very wide range of environments across the world. And obviously, if those environments are terribly, terribly poor and we're starved, we heard a bit about the Dutch hunger winter earlier on this morning, then our survival and our health is poor. Equally, if we live in a very, very rich environment, as many of us these days do, our survival will be poor. So we have a fairly wide, healthy range. But that is predicated on the idea that our development has been optimal. We pick up signals during development which enable us to predict the environment that we'll live in. And if the signals are accurate, then we'll be able to live very healthily in that environment. But what happens if the environment's impaired in some way, in nutrition, hormonal status, exposure to chemicals, a whole range of different factors might be involved. That's going to bring the healthy range down and also shrink it, so that the range of environments that we predict we're going to live in is now smaller and poorer. And increasingly, of course, many of us live in environments as we grow up where that's not the case, and so we get an increased risk of non-communicable diseases from that unpredicted excess. Look at this picture from rural Maharashtra, which summarizes the challenge, especially in the developing world. Here's this woman. She's actually quite young. She looks rather older. She's already got 2 children. She's already got 2 children. Here she is living in this poor rural community, having to carry these calabashes of water to the home every single day. She'll always be the last person to eat in the family, and in fact, probably 2 days a week she'll be asked to fast. And even in pregnancy, she'll still be working very much on the farm. There's hubby in the background. He doesn't seem to be doing very much. That's strange, isn't it? I've never seen anything quite like that. Just imagine what will happen when one of these little chaps here decides that he would far rather live in Mumbai or New Delhi, where there are cars and streetlights and internet cafes and things like that. He's going to move from this very poor environment to a very rich environment, and the mismatched story is going to play out big time. And so it's no surprise when we look at the challenge that's facing the world today. As many of you know, non-communicable diseases far outweigh communicable diseases in terms of risk. 63 to 66% of the world's population will die of these diseases. They vastly outweigh injuries too. 80% of these injuries are occurring in low and middle income countries as they go through socio-economic transitions. The mismatched story is playing out big time across the world. But we can't forget this side of it either, because this picture could have been taken in almost any city in the world. And we worry, of course, about the pattern of obesity in these 2 ladies, but we also worry about the future health of the little baby in the pushchair or the stroller here. And sure enough, if we look at the epidemiological data, this is taken from the Aberdeen study, and we do what's called a Kaplan-Meier plot. It's for survival of a population, starting with the entire population here in their early years and declining through until their 60s. You can see, I hope, that graph is that those women who are obese pre and during pregnancy have offspring who survive less long than the controls, the normal ones, the red line over here, and it's graded in between. So you'll live shorter if your mother was obese before and during pregnancy. But what's really interesting is how early in life these graphs peel apart. There really is a shadow of that developmental trajectory following us through in our lives in terms of risk. And we wonder how that can be in terms of evolutionary biology. But of course, those obese women would probably never have existed on the savannah. And indeed, the disease that many of them will suffer from, gestational diabetes, associated with high levels of glucose in the plasma, would never have been seen in our evolutionary past. So when we take the human placenta, as Priscilla Day, a student with us, did, and perfuse it with glucose on one side and measure what gets across to the fetus, there is no limit. You can go up to saturated, pretty much solutions, Coca-Cola levels of sugar on one side of the placenta, and it just gets straight across to the fetus. There was never any evolutionary advantage in evolving a placenta that protected the fetus against high levels of glucose. So this message is passed from generation to generation. Well, we heard this morning too about the Human Genome Project, and of course, when I first really started in developmental biology, finishing my PhD in 1979, we were beginning to hear about the opportunities to put everything down to genes. And of course, in the 80s and 90s, as the huge amounts of funding were assembled for the Human Genome Project, it all began to look fantastically hopeful. The trouble was, it didn't work out. And there's a lovely news piece which appears in Nature, long ago now as 2008. You can tell when Nature produces a news piece, it's obviously got to be important, written by a guy called Mayer. And he talks about the case of missing heritability. We've been talking about how these diseases are passed from generation to generation. And he says, when scientists opened up the human genome, they expected to find the genetic components of common traits and diseases. But they were nowhere to be seen. Where'd they gone? Someone's stolen them. You can see the burglar. Look, he's going out through the window here with the secret. What's going on? What have we missed? We spent all these dollars and euros and pounds. And yet, when we look at some of the common diseases, this is a recent paper looking at type 1 diabetes, just coming to, and literally tens of thousands of subjects in this consortia of consortiums. The conclusion here is genetic variance accounted for only 5.7% of the variance in type 2 diabetes susceptibility in the general population. So if we go down to the hospital in Torino and we go to the endocrinology department or the diabetology department and we say, do you mind if we just borrow 100 of your patients today and we'll take some DNA from them, a buccal scrape or some hair. Of those 100 patients who've all got type 2 diabetes, maybe 6 will turn out to have this collection of genetic mutations that are associated with risk. So why are the other 94? Why have they got type 2 diabetes? What on earth is going on? And it's questions like that that have troubled the genetics and genomics community for a long time. And so you won't be surprised to hear that the answer lies in the middle of the slide, that there are epigenetic processes involving the developmental environment and the inherited genetic code which affect the risk of disease. And we heard a little bit about bees earlier on today. It's a fantastic story, isn't it? In terms of how an epigenetic switch can produce two phenotypes that are so different. These two are sisters, but you'd be forgiven for thinking that they were different species. The queen has mouth parts that she only uses for fighting other queens to the death. Her metabolism is tuned to producing eggs. She flies perhaps once in her life, when she flies on a nuptial flight at 1 kilometre of height. All the male drones follow her, they mate in midair, great fun this, and then they die. Many parallels, I think, with aspects of human behaviour here, but there we are. And then she comes back to the hive and lays all the eggs. So that's her biology, whereas her little sister, the worker, is sterile, mouth parts for collecting pollen and nectar, metabolism for flying to the flowers and back to the hive, does a little dance to the flowers and back to the hive till she dies because she's worn out. You can switch between one and the other from the same larva in the comb here simply by feeding the protein-rich royal jelly. For a week or more, you get a queen. For less, the poor diet, you get the worker bee. And it's an epigenetic switch involving DNA methylation. And one of the DNMTs, as you can show with an siRNA technique, that we heard again about that before lunch. And we heard about Waddington too, and his epigenetic landscape, where his concept of canalization allows the little ball of cells to have a different fate as it reaches these decisions here. And I think the point about canalization was that the steepness of these walls is the thing that prevents the environment from affecting the genetic program in here. This was Waddington's concept. So you can switch between shallower walls and steeper walls and constrain development in tighter or less tight ways. One of the sets of factors which is involved in this process are the heat shock proteins. And so we decided to look at this some years ago, and we measured in a series of experiments the expression of heat shock protein 90, which is one of the wall steepening components. What we did was to give a group of rats, female rats, a 25% increase in their diet, so a real mismatch scenario for them, and we held that diet at that position as if they'd migrated to a city from poor rural areas for 3 generations. Heat shock protein was quickly downregulated and it stayed down, as if now the walls, the canalization was relaxed. And we saw effects on the methylation of one of the DNMTs, DNMT 3A, which was particularly true in the first generation, and so the message for its expression was reduced as the methylation went up. And we can put it then together in a diagram where some aspect of the developmental environment changes heat shock protein 90, changes the regulation of DNMTs, allows a change in epigenotype, which is passed to the offspring. They then become mothers, and the story may or may not repeat over several generations. So we've got an interesting wrinkle on some of the things we were hearing this morning. It's true that some of these epigenetic changes can be passed to future generations. That might be because they are literally preserved intact and passed on, or it might be because they're reinvented in each pregnancy, if you like, or in each conception, in each developmental trajectory. And I think sometimes we don't distinguish between those two processes. Probably, of course, if you're talking about epigenetics and evolution, you can't do better than go back to the absolutely archetypal species, Darwin's finches. And there's a very nice paper by Mike Skinner and his group here, where he decided to look at the different species. I think there are 5 of them here, of extant Darwin's finches. And to look at here's their cladogram here, to look to see the extent to which they're similar in terms of genetic mutations, and the numbers are shown here in blue, and there are obviously quite a few, but also in terms of epi-mutations, differentially methylated regions, quite big regions, I have to say. And there are surprising numbers of those, too, between the species. He's even been able to show that with tourism and changes in the environment in the Galapagos, Darwin's finches are changing their epigenetic profile sort of on the hoof. So we've got the beginnings of the idea that actually epigenetic change might almost lead evolution in some way. Let me now come to the question of how these things, how the epigenetic processes might relate to the human. And as our chair said, one of the things that I promised to talk to you about was biomarkers. And we wondered how we could establish some candidate biomarkers in the human. Luckily, we have some prospective cohorts, long-term epidemiological studies, where women and their families were recruited actually even before pregnancy. We have data about their diets in pregnancy, and then we're able to follow their offspring up. And luckily, at birth, the cord and the placenta of those individuals was stored. So we decided to look at the epigenetic marks in the umbilical cord, which after all is basically fetal blood vessels with a bit of Watson's jelly and some epithelial tissues added on. And to cut a long story short, this is the data that we published in the journal Diabetes a few years ago now. We've got a cohort of children here that were aged 9, and we measured their fat mass in kilograms using a DEXA, an X-ray technique that allows you to get a quantitative measure of fat in the child's body, and muscle too. And we related that fatness or thinness of the child at age 9 to the level of a whole range of epigenetic marks at birth. And this is just one. It's in a gene RXR alpha, which forms a heterodimer with vitamin D. It's involved in adipocyte differentiation, in metabolic control. The level of methylation at just one CpG in that gene is able to account for more than 25% of the variance in fat mass of those children nearly 10 years later. When we looked at this data, we thought, this is mad. Can't be right. This has got to be an artifact. And as always, I think people are beginning to find in epigenetic studies, not so true of GWAS, of course, it's critical to repeat the observations. Those of you who work in epigenetics know just how complicated the bioinformatics is and how statistical techniques can lead you up the garden path if you're not careful. So we repeated the observations in another cohort of children. These were younger children, they were only 6. First of all, we studied just 27. We found exactly the same phenomenon. So then we took all the kids that we had good DNA from and the DEXA measurements at age 6, the same thing. So this is only one part of the genome out of millions, literally, of possible sites where this change could occur. But in terms of a proof of principle that an epigenetic change measured at birth can give a far stronger biomarker signal than anything else we know about, it really does change the game. Well, we know quite a lot about these women, and one of the things that we know about is their diets in pregnancy. And many women in Southampton, and I would say here as well, and in many other parts of Europe, are dieting before pregnancy and even in pregnancy. They very often try to indulge in a sort of Atkins diet where they think, oh, carbs are bad, mustn't have any of the pasta, mustn't have any of the rice, mustn't have any of the bread. And they try and make up for it with protein, but it's very often not high quality protein. It's not nice red meat or oily fish. It's cheese and other forms of dairy protein. And that combination of low carb and high protein actually is associated with the epigenetic change in the child's cord that we've just seen. Not such clear data, it looks as if there's a sort of threshold effect here. But remember that that unbalanced diet then is associated with changes in the epigenetic status of the child's gene, and that increased methylation is associated with greater fatness in the child. And these things were independent of mother's body mass index or indeed of birth weight. Trouble with those studies is that, of course, they're using the cord, which is not a tissue that's necessarily readily available. And so colleagues of mine in Southampton decided to look at another cohort of children where fat mass was measured as they grew up, here between the ages of 9 and 14. These are boys and girls. And they're looking at another gene also involved in adipocyte differentiation and metabolic control, PGC1 alpha, the peroxisome proliferator activated receptor gamma, coactivator 1A promoter, lovely name, PGC1 alpha to its friends. And they looked at 4 CPGs in the promoter region of that gene, measured at 5 to 7 years in the blood of these children. So now we've got a very accessible tissue. And they found, and here's the mean and standard deviation for each of the CPGs, that not only did it predict adiposity in the children, but it tracked throughout childhood. So again, only proof of principle, one single example, but now we're getting to the situation where we may have some biomarkers that actually track with a life course condition as these children get fatter or thinner as they age, and might therefore be used to track the efficacy of interventions. We heard about this funny little organism earlier on today. I was brought up to think there's no way that any epigenetic change could be passed from one generation to the next via this, that all the DNA was impacted into protamine, that all the epigenetic marks were wiped and so on, and anyway, there was no plasma in the head of the sperm which could contain any of the small non-coding RNAs that we were hearing about just before lunch. All those things are wrong, and there's lots of lovely data. Here's just one example coming out of the animal and indeed the human literature, that smoking in fathers induces changes in microRNA expression in human sperm, and that some of the changes are associated with metabolic control genes in the offspring. So now we've got some great biology that it's not only the mother's status before conception or at conception and through pregnancy that matters, but the fathers as well. And that, to my mind, is another game changer, because sure enough, if we don't engage the blokes in this story and just blame women for the whole thing, which is something that has begun to happen, we will lose any opportunities to promote global health. I wanted to give you a bit of history. Some history that we didn't hear this morning, I don't think. My Italian's not very good. Not very often discussed. That actually, up until about the time of the Second World War, much of what we've talked about, not the detail of the molecular epigenetics, but some of the principles, were very well known actually in the former Soviet Union, in Russia. And here is a wonderful guy, Igor Shmuelhausen, who wrote a wonderful book on the dimensions of evolution with his teacher, Sevastsos. They had the concept that development was very important, that the environment could affect it, and that you could have different phenotypes which might adapt in different ways according to that story. It got lost because their story was captured, stolen by this guy, Lysenko, who was completely fraudulent. He had ideas that if you planted trees closer together, they wouldn't compete because they were the same species. And of course they all died. He had ideas that you could manipulate fruit trees to get much more fruit on them to grow, and the trees didn't do that at all. He had ideas that in fact if you cooled grain down, you could actually get potatoes to grow twice a year, get 2 crops of potatoes, 2 crops of wheat, which of course would have been fantastic for the Russian industry, for Russian agriculture. These ideas, though, were taken by some of which had basis in fact, most of them were fiction, were very much taken over by this guy. And here he is, not addressing the Torino Academy of Sciences, but the Russian Academy of Sciences. Can anyone tell me who's in the chair? Yeah, Stalin. So here's Lysenko, very much under the patronage of Stalin. Stalin loved the idea. What could be better than to get the slaves who worked in, the peasants who worked in agriculture somehow conditioned by some of this epigenetic processing by the environment, so they would work as hard as the slaves in the Soviet factories in the cities. And so engineering the human soul, as Stalin put it, was a great project. And of course it fitted very well with the sort of bullshit, excuse my French, that Lysenko was selling him. And we know actually that Waddington, he's seen here, met Lysenko. Here they are. It's in the early 60s. There's just a short note of it in Nature where Waddington talks about it. We don't know what they discussed, and we've no idea the extent to which Waddington's ideas were influenced by Lysenko. But when I became interested in science as a kid in the 1960s, I was told, yeah, well, you know, concentrate on Jacob and Monod. This is where we're going. There is some funny stuff that came from Russia before the war. Yeah, Pavlov, he's okay, but all the rest of it, it's just rubbish. They made up their data, ignore it. That was a tragedy, and I think in many ways the fact that Waddington didn't get further and have greater acceptance of his ideas was because he was tainted by being a commie, and he may have been tainted because he'd been to Russia and had talked to people like Lysenko. So Cold War politics meant that much Russian pre-war science was ignored. Epigenetics itself was very much overlooked or thought just to be soft, unscientific or associated with communism in some way. The gene-centric view dominated until, of course, it was rediscovered really in the final years of the 20th century. Now we know that it has enormous relevance. Just lastly, a couple of points. One, I think, relates to how we might apply this to public health more broadly. And I think it's very interesting if you compare two models of what you do with the limited resources that you've got to developing young people. Do you take the so-called luck egalitarian view, often credited to Hanson, which is basically that you should give greater resources to those people who have a poor start in life through no fault of their own. Yeah, their mums had gestational diabetes. They came from a poor background, poor diet, father smoked before pregnancy and so on. Do you say, well, okay, these are the people who should have greater resources? Or do you take the Rawlsian view that, no, that's not what, we don't worry about any of that stuff. Let's take people at a time when we can measure them, probably in their early lives, and say, right, well, these people have got great potential, but things are holding them back. Never mind why some have got more potential than others. These people have got great potential. Let's give them more resources. I think that the epigenetic story, developmental epigenetics in particular, makes this whole thing, and it's linked to the nature-nurture divide. And again, if you want information on that, read Evelyn Fox Keller, The Mirage of a Barrier Between Nature and Nurture. It destroys it. There is no distinction between nature and nurture. They're inextricably wrapped up through epigenetics. And so we might well have every reason just to invest in a good start to life for everybody, regardless of how they've got to where they are. We heard a lot actually, and I'm no expert, about the passage of epigenetic change from one generation to another. Jablonka and Lamb wrote about this in the 1980s, and it's been very much criticised. There's a good debate on this in the International Journal of Epidemiology. This is one of Emma Whitelaw's papers attacking them, and it's well worth looking at if you're interested in this area. And so Emma here is saying that here's an environmental stimulus which produces a change in the gene's expression state, and that might then be passed to the next generation. But what happens if it produces a change in a germline? Then of course it can indeed be passed on, and that then might be passed to 2 or more subsequent generations. So we genuinely can have epigenetic inheritance of risk. But you can take it further and say, well, so we've got an environmental change, we've got an epigenetic response, which, as I said earlier on, is good old-fashioned physiology, that then may be passed to the next generation by this soft inheritance thing. That can produce a new phenotype, which, if it's adaptive, will survive. Now, if the environmental change is sustained, something else can happen. Maybe we don't have to wait multiple generations for a lucky genetic change, a mutation to occur, which will confer survival advantage. Maybe that epigenetic change itself can protect certain parts of the genome from spontaneous mutation and open up others to allow a greater chance of mutation. And that's exactly what new data is showing. And of course, it can now be targeted. So we can have directed evolution, and we can have evolution occurring over just a few generations rather than thousands. And we can even have evolution running backwards, that the environment somehow drives the genetic change which is heritable rather than the genetic change leading to individuals who survive or don't in an environment. And that's absolute heresy, and it's the sort of thing that could really get people burnt at the stake. And can I smell burning? I'd better stop. Thanks for listening. We don't want questions. Do we want questions now? No.
How do you prefer? Do we have some questions now? Okay. I think we can just
thanks a lot for this talk, very interesting. I would like, of course, to challenge your heresy, not to burn you, though, on a stick. Anyway, one might say that the problem with this view, the model you have presented, in terms of the adaptation of some mediated through epigenetic mechanisms to the environment, which then becomes a trait, actually conflates two different mechanisms. One is adaptation, the other one is proper evolution. One could tell you, indeed the epigenetic mechanism which allows us to get programmed for a given environment after development, so at birth, is a mechanism that has been selected through natural selection, so it's a Darwinian mechanism, it's genetic. There are, it's anyway proteins and genes that code for that, and indeed we have this device which allows us to adapt to the environment. Then the selection, the proper evolution point would come only when actually you have the genetic mutation that is incorporated and passed through generations. So in your flowchart, so to say, inheritance and evolution would properly come only when the mutations get incorporated into the genome and passed to future generations, rather than when you acquire the epigenetically acquired trait.
That's because I was trying to be as heretical as possible. I was still taking the tenet that, excuse me, that the inheritance does, at the end of the day, have some purely genetic component. There's no reason why it shouldn't be an epigenetic component that's sustained for multiple generations, possibly forever. Certainly the animal data with some endocrine disruptors shows up to 5 generations via the male line. I suspect that that's going to become expensive. So from a parsimonious point of view, it probably makes more sense to use this as a way of exposing bits of the relevant bits of the genome to the potential to mutate. So then you have a cheaper fixed modification to the production line, and so the genetic effect is there. What was the other thing that I was thinking, though, as you were asking that question? It's gone. It'll come back in a minute.
Can I just reply? Maybe you will remember.
Yeah.
But that's, I definitely agree that probably we should revise the idea of having just evolution through genetic traits. So this is completely also in tune with, I was thinking about while you were speaking, one of the papers in the special issue for the publication of the Roadmap Epigenomic Consortium data, which was showing that patterns of histone methylation and epigenetic modifications at different levels are actually predictive of mutations per cell type, meaning that that particular epigenetic patterning in a cell, that cell type will give you a hint about what kind of mutations you're going to find in that particular type of cancer. So that's definitely something that...
It's definitely, yes, yeah. I think the other thing I was going to say was that we mustn't, a lot of the interest these days is in genetic, epigenetic interactions. We find, for example, in another cohort in Singapore, that if you do a GWAS study of the 3 ethnic groups there, Indian, Malay, and Chinese, you can separate them by their genotype, of course. If you do an epigenotype, you can't separate them at all. But then if you do a principal component analysis of the epigenotype in relation to, say, mother's response to stress in pregnancy, postnatal depression, her diet, then they separate. So actually, there are genetic factors which interact with epigenetic mechanisms to produce phenotype and vice versa. So I think we're left perhaps with Evelyn Fox-Keller's point about the genes being the followers in evolution, not the leaders. And Fox-Keller would go further and say, genes actually don't exist. They're just a concept. There is no thing called that actually which you can home in and say that's a gene. It's only a gene when it does a certain job, when the transcription factor comes along and all the right cocktail of non-coding RNAs and other things are there. And then, of course, there's the expression. And that can change. It may involve different bits of the DNA. There's a huge amount of interaction. So it's a nice concept, but I'm beginning to think it's had its day. But again, that's heresy. Thank you.